Thank you. Hello, everybody. This is part 2 now of the transporting plants lesson, if you like. So just to cover what we have gone through in part 1, we talked about vascular tissue, the structure and the function of xylem, largely xylem. We did a little bit about phloem, but that's something we're going to come on to today. And the key thing we looked at last time was the pathways of water and minerals from the soil into the xylem. Those recordings are available now, so you can go and look at those if you haven't done so already, so that you can then be in a better position to engage with what we cover today. And what we cover today will be, we intend to look at transpiration. Now that we've talked about the movement of water into the plant, we're going to look at the pathways of water through the plant and then the loss of water from the leaves in transpiration. We're then going to look at adaptations of plants. So looking at the environment those plants live and then the adaptations that they have to cope with those environments. And then finally, we'll look in a bit more detail at the structure of phloem and look at the mass flow hypothesis. So let's get through as much of this as we can in the time we've got available so that we can cover what we need to. So let's go. Next. The first thing I want to show you, shows you an activity that I think is always a good thing to look at. It says, add to the diagram given, which shows a simplified root, stem and leaf, and show how the transpiration stream works, annotated with as much detail as possible, including the forces involved and how they arise. So let's just look what we've got here. So what I want to do is just show you, explain to you what those forces are involved. I'm going to show you how this should look, and then we'll do a little activity for you to sort of gauge your own recall and understanding on that. So we've got a very simplified situation here. We've got a root down here where we've got some cells. We've got xylem vessel in the middle. We've got some cells, which are largely going to be parenchyma, and then we've got a root hair cell on the end, and we've got soil and soil water here. And then at the top of this very simplified plant, we have the leaf, and it shows some leaf cells here. And importantly, it shows a stomata present as well within the leaf, which we'll come on to as being that location from which water vapor is lost from the plant. And then these blue dots here are meant to signify water. So here's the key. I've got water molecule that will travel up through the xylem. And then I'm going to show some forces involved here, and I'm going to show the movement of water too. So first thing is that we know that water vapor, transpiration is by definition the loss of water vapor through the stomata, from the leaves through the stomata. And this loss of water vapor is a result of evaporation. So we'll come on to this a little bit more, but the principle or the reason behind stomata being present is so that gas exchange can take place, for carbon dioxide to come into the leaf for photosynthesis, and the oxygen that's produced in photosynthesis to leave the leaf. However, the unfortunate consequence of having stomata is that water vapor will be lost. Now, that is not desirable. Plants don't want to lose water vapor, but they have no choice really, because they have stomata, and that's going to result in the losing water vapor. And they do then utilize the fact that water vapor is lost to draw water up through the xylem. So how does that happen? So because water is lost then from the leaf, water is replaced from the xylem. Now, going back to what we covered last time, we've got those three pathways, the apoplast, symplast, and vacuolar pathways. Those are still employed, those are still utilized in the cells of the leaf. We always talk about them happening at the root, but they happen in the leaf too. So water is drawn in from the xylem through those pathways. Now, what that does is it draws water up the xylem vessel from the root to the leaf. Well, why does that happen? Why does water moving in to the leaf cause water vapor? Water being moving into the leaf cause water to be drawn up through the xylem. So the reason for that is because we've got forces involved. Now, I'm just going to come back a little bit and say, before we do that, look down here. We've also got water drawn up from the xylem vessel, and then down the bottom we said water moves into xylem through the apoplast, symplast, and vacuolar pathways. So in the same way that as water is moving into the leaf and moving through the leaf cells through apoplast, symplast, and vacuolar pathways in the root, the same thing is happening. Water's moving into the xylem via those those same pathways. But we just need to now justify why it is we're getting, what forces are involved. So what I've shown on there is just the movement of water. Water's lost through stomata, it's replaced from the xylem, it's drawn up through the xylem, and it moves into the xylem at the root. Okay, so what forces have we got going on here? Well, the first one is this force we got called root pressure. So because water is being drawn in to the xylem, you get a buildup of pressure inside there, and that gives a sort of upward motion, then an upward force of forcing that water towards the leaf. It's not all that significant, but it does contribute. And this is perhaps, it would be lovely if we could just say there's one force involved here, but actually there's multiple forces involved here, which causes that water to move up through the stem. The next one is cohesion, okay, or cohesive forces. Now, if you remember in Unit 1, water is a polar molecule, which means it's an uneven distribution of charge, and there's a hydrogen bonds which will form between adjacent water molecules. Individually they're weak, but collectively they're very small, and collectively those forces, we call them cohesion. Now what those cohesive forces do, and in fact this we mentioned in Unit 1, is it allows tall columns of water to be drawn up the stem of a plant. So when a water molecule moves into the leaf, the hydrogen bond that exists between it and its neighboring water molecules draws up the next water molecule, and the same thing happens. These water molecules draw up from one to the next. So you can see then, there we are. So you can see that when this water molecule here moves in, it's going to draw up the next water molecule, which draws up the next water molecule, and any water molecules to which they are bonded through these cohesive forces. Now, there are also adhesive forces, and that's because the lining of the stem is hydrophilic, meaning it likes water. Consequently, we get adhesive forces between the water molecules and this hydrophilic lining. So that generates a slight upward force too. So we've got three forces working right now. We've got root pressure is a force exerted from the root pushing upwards. We've got cohesive forces, which is that drawing up, that tension that's drawing up water molecules into the leaf, and then adhesive forces, the forces between the hydrophilic lining and the water molecules themselves. But there's more. There's also capillary action. Now, capillary action is a phenomenon found when you have very thin tubes and you put them in water. You see water rise up that tube. Now, that happens, and that's more likely to happen. It will move up higher the narrower the diameter of the tube. Xylem vessels have a reasonably narrow diameter, but it's likely that capillary action is only having a significant impact in very young plants, very young shoots. It's likely that for a mature tree, capillary action is having virtually zero effect. The main force involved in drawing water up through the plant is cohesion, but they all contribute slightly, and you need to know about all of those really. But the one they're most likely to ask you about is cohesion. And here's a past paper question, which I'm going to give you a moment to have a look at, and then we'll go through. I should say, really, I mean, with all of these, because these are recordings as well, that sensible thing to do here is to pause the recording, have a go, and then see how you get on. So let's see what we've got here. Water is absorbed into the root, mainly through root hairs. Its movement through the apoplast is prevented by, now this links back to what we did last time, by the Casparian strip in the endodermis. Movement through the symplast pathway is aided by plasmodesmata. Plasmodesmata, remember, being those bridges between adjacent cells. The water is pulled upwards by the transpiration pull, and this is possible by large cohesion forces. It says cohesion. Cohesive makes more sense, doesn't it? Cohesive forces between the water molecules, and adhesion, and it should be adhesive forces between the water molecules and the hydrophilic lining of the cells. So there's a example of past paper question, largely recall, quite low level, but it gives you an idea. Whenever you do a past paper question, it gives you an idea of the where they're sort of pitching the their questions and the sort of thing they like to focus on. Right, so let's have a look at transpiration in a little bit more detail. So first of all, I mentioned this. Transpiration is the evaporation of water from inside the leaves through the stomata. Now, there's a leaf. You see the stomata on there, and you can see water coming out. Mainly find them on the underside of the leaf. This is something you cover a little bit more in the gas exchange unit. We're really more concerned about the transport aspect of this. So let's focus on that more than anything else. But we do need to have a little think about what that means, what transpiration, how that results, how it's affected. So hopefully you can see this video appearing here now, which is not especially important with regards to the content, but you can see the transpiration taking place across that in that rainforest. You can see the water vapor leaving those trees, and that is a consequence of the huge amounts of transpiration that take place particularly in a rainforest where temperatures are high. Now, stomata need to be open during the day to allow for gas exchange. Okay, and we talked about that. The reason that stomata are present is to allow gas exchange to take place, oxygen to be allowed out, and carbon dioxide to come in. But the negative side to that is that when you've got these pores, when you've got these stomata in the leaf, it means that valuable water is lost from the plant. Now, we're going to look at factors which affect transpiration. I've mentioned here, I've shown you what we just referred to, what we've drawn on. There's the transpiration stream, that activity I just went through, where you've got the loss of water from the leaves, and then water's drawn up through the stem to replace that, and water's drawn in into the xylem of the root to replace any water that's been taken up through the xylem. And we talked about the forces that were involved in doing that as well. Now, we need to look at what affects transpiration and what we call the transpiration rate, which is the rate at which water is lost from plants. Now, it's determined by a number of factors, and the three we're going to focus on are temperature, humidity, and air movement. You could also add a light intensity to that, something that covered more in the gas exchange unit, because the mechanism of stomatal opening is needed there. But we're going to focus on these three here. So why is it relevant? Well, look, what we're really interested in here is the concentration gradient between inside the leaf and outside the leaf. So when we talk about inside the leaf, we're talking about the concentration of water vapor molecules here, and outside we're talking about the concentration of water vapor molecules there, because it's the rate at the rate of transpiration is dictated by the difference in concentration between inside and outside. And so any factor that increases the water concentration gradient between the water vapor in the leaf and the surrounding atmosphere increases the rate of transpiration. So that's how we determine whether the factors I've mentioned there, temperature, humidity, and air movement, affect the transpiration rate. So let's have a look at another activity that I can use to model these differences. So I said draw a bar chart to show the concentration of water vapor inside and outside a leaf, which results in transpiration on a normal day. Then show how the features which affect transpiration affect that graph, and subsequently the rate of transpiration. That might seem like a complicated task, but look, I'm going to show you what's going on here. I'm going to sort of answer this or respond to this activity so you can see what I'm talking about. So look, here we've got, I'm showing you what's happening with temperature. Okay, so here's my starting point here. I'm showing you inside the leaf here and outside the leaf here. And we can ignore the fact that this is a high temperature at the moment. Just appreciate that I've got more water vapor molecules inside the leaf than I have outside the leaf. Now, here's a bar chart which would show that. Now, if you come over to this side, so this is like our starting point, our normal situation, where you can see inside the leaf, I've got more water vapor molecules than outside the leaf. And as a consequence, we're going to get a movement of water vapor from inside the leaf to outside the leaf. But what happens when you increase the temperature is that you increase the evaporation that takes place inside the leaf, and consequently you can end up with a higher concentration of water vapor molecules inside the leaf. So we've increased the number of water vapor molecules inside the leaf. Outside the leaf hasn't changed. Okay, that stayed the same. But what we now have here is a bigger difference between inside the leaf and outside the leaf. And that means we're going to get a greater rate of transpiration, a greater diffusion of those water vapor molecules from inside the leaf to outside the leaf. Now, note I say diffusion, I don't say osmosis. Okay, we're talking about water vapor molecules, gaseous water. So we're not talking about osmosis, where we need liquid water if we're going to be referring to the movement of water. Here we've got water vapor, so it's diffusion we're talking about. Now, if we contrast that with a low temperature, how would that graph look? Well, look, you can see what's happened here now is that there's a big drop in the concentration of water vapor inside the leaf now, because there's much less evaporation going on because the temperature is lower. Temperature affects evaporation more than anything else. You might also say it affects the kinetic energy of the water vapor molecules, and therefore they will move faster and diffuse faster. And that's a relevant point. But primarily we would talk about a greater level of evaporation and therefore a greater concentration when you have a high temperature. Now, looking at humidity, and certainly humidity is just a different take on what we've just shown. Air movement is a little bit different, but let's go to humidity. So now you can see I've got this little key over here which says in pink is what's been affected. So here is what's been affected, this pink thing out here. Okay, and look at the difference between this situation compared to it was with the temperature or the conditions outside the leaf when we were looking at temperature. You can see there's a higher concentration now outside. Inside hasn't changed. It's meant to be the same. It's meant to be what it was before, but outside has changed. So look what's happened. The concentration outside the leaf has increased because it's very humid. It's high humidity. That means lots of water vapor molecules are in the surrounding environment. So there's only a very little difference between inside and outside the leaf. So we're not going to get much diffusion happening of those water vapor molecules. But if we look at low humidity, well, now we've got a much lower level of humidity outside the leaf, a bigger difference between inside and out, and so we're going to get a greater movement of water vapor molecules out of the leaf. What about air movement? So air movement is a little bit different. Air movement is all about maintaining a concentration gradient. So what's being shown here is you've got water vapor molecules which are moving out, but then as soon as they move out, those water vapor molecules are being blown away because we're talking about high air movement or lots of wind, that's saying. So what it's doing is the water vapor molecules are diffusing out and it's increasing the concentration outside, but they're immediately being taken away by the wind, and so it establishes or maintains a low concentration outside of the leaf. If we contrast that to low air movement, so it's not a very still day, well, water vapor molecules move out, but then they stick around outside. They're not taking away. The wind doesn't take them away, and therefore they stay in the locality. They stay there, and so you don't get as much diffusion moving. You do get some, but not as much as when we had a high air movement. We get lots more diffusion out. Right, so based on what I've just said, this is a great opportunity to pause the video and come back and have a look, see if you can answer these questions. So I got here, transpiration is it's the evaporation of water from the leaves of a plant. This occurs down a water potential gradient through stomata. A high temperature would increase transpiration as there is increased evaporation and therefore an increased diffusion gradient. You could have said concentration gradient between inside and outside the leaf. There is also an increase in kinetic energy, sort of an additional point with temperature that I mentioned. So there's an increased rate of movement of water molecules. Increased air movement increases transpiration as it maintains the concentration gradient between inside and outside the leaf. Whereas high humidity decreases transpiration as there's a decreased diffusion gradient between inside and outside the leaf. And this is a high light intensity increases the rate of transpiration as it causes stomatal opening. Now, that's not something I've referred to yet. That's more in the gas exchange unit, but it's obviously still relevant. I mentioned here about the use of a potometer here, not something I've mentioned here, but it's worth, we'll read through it now. Potometer is set up underwater to ensure that air cannot get into the apparatus. The leafy shoot can be exposed to a change in environment, such as increased air movement using a fan. And the volume of water taken up per unit time, as shown by the bubble of movement of the bubble, will give a measure of water uptake by the plant. This will give an indication of the transpiration rate. I haven't mentioned more about the potometer here. It's something you see a lot in past paper questions, and we're going to have a look at one of those now. So first thing, it says table shows the transpiration rate of a plant measured at regular intervals over a 20-hour period. Plot these data on the graph paper provided. Now, obviously, this is not something you can do, but I have actually done this to show you just some key things about when you're plotting a graph. So just make sure that you are putting the whatever's written in the table into your labels on the axes. I've put the times on. I put the rate on at the side. Just make sure, key thing here, you have a value at the origins. Both origins should have a value there. They does not have to be 0, but you must have something there. It's probably the most common mistake seen on graphs is that there's no value at the origin. Okay, and then plots. Well, I'm not going to do this. This is difficult for me to do on a computer to show this, but plotting is something that pupils generally don't have an issue with. And then remember, you've got to join together with a ruler, which have I shown on there? There we are. I've shown on there. And it says describe and explain the difference in the transpiration rate at 0800 hours compared to 1600 hours. Now, the key thing here, I mean, I'm going to just take that away a moment. The key thing here, I would be worried about the fact that this is 4 marks. So it's a describe and explain. I think, well, what can I say to that? So look, what's on there? Well, they wanted a quantitative comparison using numbers to, rather than just saying it's more at 1600 than 0800, you actually have to use values there. Then second mark for mention the difference in light intensity of the 2 times, which links closely to what I said about the gas exchange unit, which again is linking to, again, we haven't got over it here, but potassium pump and causing stomatal opening. I won't mention any more about that because that's at a different unit, but I just wanted to show you that because it was worth seeing the graph. Right, this is more relevant to what we're talking about under experimental conditions. So again, this is an opportunity for you to pause and you can look at the answers then when you've had a go at these. So under experimental conditions, give 2 ways in which the peak value could be increased. We can increase the temperature, we could lower the humidity, or we could alter the air movement. So we could increase the air movement, couldn't we? The cohesion tension theory explains the movement of water up the xylem of the plant. What is meant by cohesion? That we talk about that attraction between water molecules. How tension is created? Well, the evaporation of water molecules through the stomata, or transpiration at the top, is replaced by water coming in from below, in from the xylem. That creates that tension, that draws that water up from beneath. What additional force helps to support the water molecules in the xylem against the force of gravity? Oh, sorry, I didn't finish that. There we are. Oh, of course, because that's 2 marks. Because of cohesion, this creates an upward force through the whole column. So, right. Part 3, what additional force helps to support water molecules? Adhesion. Okay, we talked about that. And the values plotted in the graph never fall to 0. This suggests that throughout the 24-hour period, a constant additional small force is influencing the upward movement of water. So what is that force? It's root pressure. Now, you might have said capillary action, but remember that's very much on the sort of the bottom answer. More relevant in shoots. We haven't mentioned root pressure there. How is this generated? Active transport of ions into the root xylem from last week creates an osmotic gradient, or water is drawn in by osmosis. That's what we mentioned last week. Okay, right. let's move on. I'm conscious of time. You can always pause and go back and look at these. Here we are now. So we are covered now transpiration. So let's go on to the adaptations of plants according to their environment. So here, similar, we have an activity where I asked to compare the features of hydrophytes, xerophytes, and mesophytes. So hydrophytes are plants that live in a watery environment. So they're happy living in an environment which has got lots of water around it. Something like a water lily would be a perfect example. Xerophytes, they live in an environment which is very dry, something like marram grass. You might things that would live in sort of very dry environments, such as like a desert conditions. And mesophytes are the ones that are sort of in the middle, and those are the plants that we're used to in our temperate climate in the UK. Right, so this is what a hydrophyte would look like through a microscope, cross section of the leaf, and you can see. Well, let's have a look. What feature would we have? First of all, we don't see any lignified tissue. Now, lignin is there as a support. It acts as a supporting tissue. Well, hydrophytes don't need lignin. Because they've got water. Think of a water lily. It is sitting on the surface of the water, so the water is acting as its support. So it doesn't need lignification. It doesn't need to expend energy in lignifying. So it doesn't have it at all. There's very little xylem there. Okay, it's not very well developed. Again, there's no need for much of a transport mechanism through the plant because there's water everywhere. There's no cuticle. So a cuticle is present on the surface of the leaf to reduce evaporation of water from the leaf. Well, the water lily does not care about losing water through evaporation. So it doesn't bother, again, no point it wasting the energy on making a cuticle when it's not really serving a purpose. Stomata on the upper surface, well, think of a water lily. If the stomata are on the lower surface, they're going to be in direct contact with the water, so you're not going to get gas exchange. So they need to be on the upper surface. Now, that would mean there'd be more water loss coming out because having them on the lower surface means it reduces water loss in a normal plant, a normal leaf. Water lily, number one, couldn't have them on the lower surface, but also does not care about losing water because it's got so much of it. So having them on the upper surface is no issue. And it has large air spaces. So you can see those large air spaces here. This is the spongy mesophyll. Well, this section there is the spongy mesophyll. Look at the size of these air spaces. That's because it provides buoyancy. Okay, now if we go to a xerophyte, there's a bit more going on here. First of all, they have rolled leaves. Now I'll show you what a rolled leaf looks like. Here's an image of a rolled leaf. So this leaf started off, I say started off, you imagine it could open out, but it doesn't. It is rolled in on itself to look like that image there. So we have rolled leaves. And what that does is effectively makes the inside or the underside or this side of the leaf is now no longer exposed to the outside. Or should I show here? This side of the leaf is now no longer exposed to the outside. It sort of internalizes that leaf and reduces that total surface area that's exposed to the outside. Here's an example of why rolled leaves works. You can see that if this was a normal leaf here, you see the water vapor molecules leaves, water vapor molecules leave, and they're then in the external environment. By having a rolled leaf, those water vapor molecules stay within that sort of rolled up section, and you create this little microclimate, which is more humid, and that, just like we talked about, that reduces the diffusion gradient between inside the leaf and outside. They also have sunken stomata. Those stomata are lower in the leaf. So this is our normal leaf on the left-hand side here, where the stomata are, well, should we say normal? Now look what's happened here. We've got sunken stomata. They're set back from the surface. And again, what that does is it creates a humid environment in that little groove in there. So it means that when water blows across that leaf, we've got this little microclimate in here which reduces that diffusion gradient. They have hairs on their surface. Again, what that does is it traps water vapor and so reduces that water potential gradient. It has a thick cuticle, just as the hydrophytes didn't need a cuticle at all. Xerophytes really do, because it's minimizing transpiration or evaporation from the leaf. You might also get thick sclerenchyma. We talked about parenchyma. Sclerenchyma is like parenchyma, but with much thicker cell walls, and that provides a degree of support, so it's less likely to wilt. They have succulent stems. Classic example here is a cactus. Say the cactus stem stores water. It's not quite like that, but really it has water storing tissue within it, so that when it does rain, it can store as much water as possible because it knows there's going to be a period of drought after that. There's lots of succulent plants, house plants actually, something like aloe vera. When you squeeze it, it feels a little bit squidgy, and that's because it's got water inside it. And they may also have needle-like leaves, and that again reduces the surface area, reduces transpiration. Now mesophytes, a little bit in between and doesn't quite work in the same way. Nothing all that obvious relating to water. It's more just specialisms that they have. So the fact that they shed their leaves before winter, or perennial plants do, so because there's no photosynthesis happening during winter, so they shed their leaves. The non-woody plants will die above the ground, so they will freeze. If they stay above the ground, when the temperatures get to freezing, they will freeze and they will die. So it's best to reabsorb, for those plants to reabsorb the nutrients from those parts of the plant above the ground, if you like, store them in their roots over the winter, and then they can shoot in the spring. Shoots will appear using those stored nutrients. There's no point them being there over the winter. And the other thing, depending on the plant, some mesophytes survive winter as seeds, because this is really about types of plants, really. So perennial plants, they die above ground and sort of retreat into the ground, but annual plants, they grow each year and then they release seeds. The parent plant will die and the seeds are what survive, and those seeds just lay dormant over the winter and then they sprout again in the spring when the temperature is right. Now, worth mentioning, hydrophytes, I would say this: the very few questions on mesophytes, I can't think of any that I can't recall any. About 80% of questions, this is not an uncommon part of the specification to be examined on. I would say about 80% are asked about xerophytes and 20% on hydrophytes. So worth knowing that. Okay, let's go on. I'm going to do the same thing here, give you an opportunity to pause the video, and then we'll go through it. Okay. So what's the key words we often hear? Xerophytes have rolled leaves as a result of the thin-walled epidermal cell. I didn't mention this there, but they have these hinge cells which are responsible for allowing these leaves to hinge and become rolled. These cells shrink when they lose water from excessive transpiration and causes the leaf to roll in on itself. This reduces the leaf area from which transpiration can occur, which I didn't mention then. Xerophytes also have sunken stomata. These stomata are found in grooves on the inner side of the leaf. These grooves trap humid air immediately outside the stomata, so reducing the water potential gradient between the leaf and the outside atmosphere. This reduces the diffusion of water vapor. Xerophytes also have stiff interlocking hairs which trap water vapor, again reducing the water potential gradient. They also have a thick waxy cuticle which lowers the rate of cuticular transpiration through the cuticle, or evaporation through the cuticle. Xerophytes might also have succulent stems for water storage. Leaves reduce to spines or needles, so reducing surface area and the ability to close stomata during daylight. Now then, let's draw a line underneath that. Again, I'm conscious of time. We've got about 10 minutes, and I'd like us to have a quick look at phloem and the mass flow hypothesis. So we've done now those adaptations. We're on to phloem and the mass flow hypothesis. And the key features here, another activity that can be done where you draw an annotated picture of phloem tissue. Look, here's the key stuff that we've got on here. You can see we've got the key features here. So I've got, this is the main part responsible for transporting sugars and amino acids and solutes. You have this section here, which is referred to as a sieve tube cell or a sieve tube element. You have a number of these sieve tube elements stacked along top of each other. At the end of each sieve tube element or sieve tube cell, we have a sieve plate, which contains pores to allow the movement of these sugars or amino acids, but we'll talk about sugars more than anything else, to pass through from cell to cell. Now, the sieve tube cells have cytoplasm, but they've really lost all their other organelles. So the way they stay alive is because they will have adjacent to them a companion cell and a link to those companion cells via plasmodesmata. Now, you can think of the companion cell as having all of the organelles required to keep the sieve tube cell alive. Okay, so it allows things like ATP to move between the companion cell and the sieve tube cell in order to keep it alive. There's also parenchyma on there. There always seems to be, but really the business end of phloem is simpler than xylem, is the sieve tube cell and the companion cell. Another little activity, you can pause. Let's have a look at this. So phloem is made of sieve tubes and companion cells. Sieve tube cells lose most of their organelles, but are still alive when mature. They have perforated end walls called sieve plates that allow solutes to pass through. These solutes flow in both directions, a key difference to xylem. Xylem, you've got water and minerals moving up through the plant in one direction only. Solutes flow in both directions. Companion cells have the organelles and have all the organelles and communicate with the sieve tube cells via plasmodesmata. Excuse me. These cells are responsible for keeping sieve tube cells alive, for example, by providing ATP. Phloem also contains parenchyma, function as packing tissue. And this is now what we need to focus on, because this next bit we haven't talked about yet, but we're going to look at it in the mass flow hypothesis. So solutes are transported in the phloem from an area which we call the source, where the sugars are made during photosynthesis, to an area we call the sink, where those sugars are used. And that's where we're going to focus the last bit of our attention on here, showing what goes on inside a plant for this mass flow hypothesis to work. So it says draw a flow chart to show the sequence of events that results in sugars being transported from source to sink. Now you need to have this available. Flowcharts are great in that one thing follows on from the next. But look, let's see our setup here. So I've got my phloem here, and I've got my xylem here. Notice they're next to each other as they would be in a vascular bundle. This movement of solutes going from a source, which is our leaf, it says a source cell there, to our sink cell, which is the root. Now the sink cell can be anywhere in the plant which is using up sugars. The best example is always going to be a root cell, because a root cell is not making its own sugars through photosynthesis because it's underground. So there needs to be a mechanism of getting sugars to the sink cell. And then the forces we're going to show here is active transport in red, osmosis in blue, and then I'm just going to show a flow then of the solutes in yellow. Okay, and those are shown on there already, but we'll give a little bit of an explanation of what's going on. So let's start in the leaf, and don't really have to start there. We could start in the root cell and the sink cell, but we've got to start somewhere. So we'll start in the leaf. So leaves photosynthesize, and as a result of that, they make sugars. So that's the second part. Sugars are going to be made, and you can see those sugars. You can see those sugars in the cell now. Okay, there they are. Now those sugars get loaded into sieve tubes. They get actively transported, that's the red arrow there, into the sieve tube cells. As a consequence, that lowers the water potential inside the sieve tube cells. And as a result of that, if the water potential is lower, now we're going to get water moving in from the xylem, because water potential is lower inside the sieve tube. Okay, water potential higher in the xylem, lower in the sieve tube, so water moves in. Now what that does is it increases the hydrostatic pressure generated. You've effectively got more stuff going into a small space, and therefore it's building up pressure within there. And it says that it flows to sink, and we could probably leave that bit for a moment and say, well, why does it flow to the sink cell? It flows to the sink because of what is happening in the sink cell. The opposite is taking place in the sink cell. In the sink cell, sugars are being actively transported into that cell. As a result, that increases the water potential now inside the phloem. Because sugars are being taken out, it's going to be less concentrated, so it's going to increase the water potential inside the phloem. And as a result, water is going to move out into the xylem. So water moves out of the sieve tube into the xylem. That reduces the hydrostatic pressure in this area here. So it's lower in that area. It is higher by the source cell. And because you've got high pressure by the source cell and low pressure by the sink cell, we get this flow of those sugars, or that mass flow of sugars, amino acids, a solution they're dissolved in, moves from source to sink. Now that flow chart is well worth taking some time to process for yourselves and to learn, but I'm going to give one more slide I'm going to show you to just to try and help with some of those key things there. So again, you can pause. So we've got solutes transported. So you can pause this and do it in your own time, but I'll go through it now. Solutes are transported in the phloem from a source where the sugars are made during photosynthesis to a sink where those sugars are used. We've said that before. At the source, sugars are loaded into the sieve tube cells, causing the water potential to fall, to decrease. As a result, water moves into the sieve tube via osmosis from the xylem, increasing the hydrostatic pressure within the sieve tube cell. Meanwhile, at the sink, sugars move into the cells, increasing the water potential. We're talking about opposites here now within the sieve tube cell, and so water moves out into xylem, decreasing the hydrostatic pressure. Sugars then move from the region of high hydrostatic pressure, the source, to the regions of low hydrostatic pressure, the sink. So I hope that we are now confident that we have covered everything on there. It's a big topic. There's lots to be covered. And I've gone through it, I think, as quick as I possibly can, whilst hopefully allowing you to appreciate the main points, and hopefully giving you some ideas as to what you can be doing to improve your understanding, a way of processing your own notes, making your own notes to enhance your understanding, so you can then equip yourself to attempt those past paper questions that you're going to need to do in preparation for your test. So hope that was useful. I'll see you next time.